we finished with the notion of a number of enzymes that do not make any sense so far. So before we get to the point where they will start making sense, I want to point your attention to the fact that replication is semi-conservative. What does that mean? It doesn't mean that it's like half Republican, half Democratic. It means that each new copy, new double-strand DNA consists of one old, which is blue, and one new, which is red strand. Does that make sense? Okay, so you see, when strand, the blue one, the old one, is being copied, the new strand is produced. So each of those double-stranded molecules of DNA consists of one old and one new strand, which, when I show it to you here, sounds pretty reasonable, right? But believe me, it wasn't the determined fact in like early 50s or mid 50s. I don't remember whose experiment it was, maybe Hershey, maybe somebody else. But it was actually determined because there were ideas maybe the new strand is entirely new, okay, and the old strand is just old. Now that's one thing. Semi-conservative replication. Where does it start? It starts at so-called origin of replication. Eukaryotes usually have multiple origins. Prokaryotes have one per circular chromosome, and that origin of replication is AT reach. Any reason why there are many? Yes. Easy to break apart. Yes, easier to unzip. Okay, that makes sense? Now, the bacterial chromosome is much smaller than the chromosome of eukaryotes, so one origin is enough. The replication goes in both ways. Think about, if we think about zipper, imagine that you have those running things, I don't even know the name for them, okay, that go in both directions, but up and down, to unzip something. Does that make sense? Like double zipper. Okay. So it goes in both directions. It makes, it still makes one copy, but it makes it simultaneously in both directions. Okay. Like when you start, if you take a book, and instead of copying it from the first page to the last page, you take two people, and one copies from page 100 to page 1, and another from page 100 to page 200. So you split it. Does that make sense? Okay. You can see here the origins of replication in eukaryotic cells, the multiple origins. And it makes it really efficient, but look, if you have 3 billion nucleotides, you gotta make it efficient. Otherwise, you just won't be able to replicate stuff. Does that make sense? Behold. That's replication, grandeur scheme. When I'm going to talk about it, we're going to spend a lot of time on this right now. My only request for you, if you don't get something, if I'm going too fast, if you have a question, please ask me. So I think this is more complicated than gene expression in terms of the mechanics of the process. And I cannot do any plays or sketches like we did with translation. I just don't have the concept how to do that. Okay? So let's figure out, first of all, where is the DNA? The DNA is those blue strands. Okay? You see them? DNA strands? We got it. We're going to step by step. So first thing first, we need to unzip it. This is helicase that unzips it. You got it? Question? There you go. I was about to say. But we can't unzip DNA if it's supercoiled. So even before helicase, there is DNA gyrase that uncoils the DNA. Does that make sense? So we got it untangled, uncoiled, we got it unzipped. Those single strands, what they want to do right away? Go back together. So you got your SSBP here. Those molecules of single strand binding proteins keep individual strands of DNA from going back together. Single strand. Okay? Does that make sense so far? Now, let's get to the actual replication. The enzyme that copies DNA is DNA polymerase 3. It may sound counterintuitive that it's 3, but they were named in order they were discovered. So this one was discovered third. Okay? So DNA polymerase 3 copying. Does that make sense? Now this enzyme can read the DNA molecule, this strand, from 3 to 5 prime. Right? 3 to 5 prime. And that's actually very important. That's a unique feature. That's an interesting feature. So it means that here's your one strand, okay, that goes, here's 5 prime, here's 3 prime. So this enzyme, a few seconds ago, and you have to understand, this is a snapshot. Does that make sense? This is a snapshot of the process. That's like a photograph. It's not a dynamic picture, it's a static picture here. So say a few seconds ago, that DNA polymerase that you can see right here was somewhere here. Does that make sense? So DNA polymerase, wait, what did I just do? It is moving in this direction. Does that make sense? Because it reads the old DNA strand from 3 prime to 5 prime. Like we read the book from left to right, from page 1 to page 100. Does that make sense? Okay? We good? Is that clear? That's simple. That's simple. Now this strand, bless you. Now this strand is called leading, which means the replication process, the copying of this strand, is uninterrupted. Like you are reading the book. You start on page 1 and you can read without any interruption to the very last page. We good? Now, you may ask, what about the yes?
Why is that one leading?
Name, just called. We get there. We get there. Top 1 is leading. The strand that is read in continuous manner, you see, there are no interruptions. You see, no interruptions, absolutely not. You got it? That's why it is leading. No interruptions. I'm going to put it here. Okay? That's leading strand. That makes sense so far? Awesome. You may ask, where's the primer? It should have. DNA polymerase 3 cannot just sit on the DNA and start replicate. it needs a primer. Well, it probably was somewhere, I don't know, here. Okay? We just don't see it on this particular snapshot. Okay? You will see too many primers. You will wish that you will see less primers. Okay? Leading strand is simple. Now what about the lagging strand? Here's the tricky part. This entire thing, which is called the replication fork, according to the picture, where does it go? The replication fork, where does it move? Like it's unzipping in which direction? Yeah, to the left, yes, to the left, right? So leading strand goes from right to left, from 3' to 5' without any problem. Now take a glance on the second strand of the DNA. Where is 5' end? To the right or to the left? To the 5'. To the right. It's right here. On the old strand, right? And the 3' is on the left. So DNA polymerase, which direction should it go then? To the right. But it can't, because the entire fork moves to the left. What should we do? Does that make sense, the idea? The conundrum that we see here, the problem? The fork moves to the left, and leading strand is synthesized perfectly fine. But reading the second strand, DNA polymerase simply cannot read from 5' to 3'. It has to go in this direction. Here's what happens. Primase, the enzyme primase, makes the primer right here. You see that primer that I just circled? The primer. DNA polymerase binds to the primer and synthesizes a short fragment. Does that make sense? And it stops and jumps back. That's another primer, okay? It's gonna synthesize another fragment, jump back. Imagine that you're reading the book, not from page 1 to 100, but you're reading the book from page 90 to 100, 80 to 90, 70 to 80, 60 to 70. Yes?
Free floating, or do they get put back together?
Explain.
So there's different segments that are being replicated, right?
Yes. Go ahead. Go ahead.
So does it sort of like float and then like it finishes the next one, they attach, or does it somehow have a continuous strand?
I think I understand your question. I will answer this, and you will tell me if I answered actually the question that you asked. Just a second. So, DNA polymerase, when floating, is the DNA polymerase floating? Every time, and then you tell me, every time it reaches the previous fragment, it falls off. Is it going to be the same molecule that will reattach here? Not necessarily. The fragments, are they free floating? No, they remain stuck with the DNA, with the original DNA strand. So far, is that what you were asking?
Yeah. So.
Yes.
So it's not like let me noodle on that, but let me think about it for a minute.
Your question.
Oh, I was just gonna say DNA ligase connects the.
We get there.
Fragments.
We get there. Yes.
I was gonna ask his question.
Yeah.
Also keep the
let me finish. I think I will answer your question in a second. Okay? When I will finish with the thing, you will tell me if you have more questions. Okay? So far what we've got is that DNA polymerase that synthesizes lagging strand has to sort of run backwards in opposite direction, okay? Run this direction. Let me get rid of all that crap over here. Okay? It has to run in the opposite direction. Okay? And then jump forward to run in the opposite direction again. Okay? So the lagging strand is not one huge long copy. It consists of fairly short fragments of the DNA called Okazaki fragments that are not connected to each other. For example, like imagine that you have a train in which all cars are connected, and then by some magic button you break all the connections. Cars don't go anywhere. Train still appears as being one train, but cars aren't connected anymore. Okay? So you have to press another magic button to connect those cars together. Does that make sense? So ligase, that's the finishing touch. Ligase, DNA ligase right here, it connects Okazaki fragments. So the lagging strand becomes one long whole copy. Go ahead. Okazaki fragments. Oh, absolutely. Yeah, it's entire sequence. It's entire sequence. It just copies everything. When replication copies everything, it does not distinguish between introns and exons. Give me 1 second. I will try to picture it using colors. So imagine this. I'm doing very, very schematically. So this is your lagging strand. Okay? The black one on top. The green are primers. And the red is going to be the new DNA synthesized. Okay? You see them? So I try to make it as good as I can to show that the primer and the new DNA are one molecule. A bit of RNA, a bit of DNA, but it's one molecule. Okay? But I also try to show that here there's a little bit of a gap. Can you see that gap? Now, when DNA polymerase 1 that replaces primers comes over and replaces them, sorry, I will correct it in a second. We have two Okazaki fragments, but we still have the initial gap. This gap is not a missing nucleotide. It's a pretty much missing link between phosphate residue and 5' hydroxyl group of a sugar. They're just not connected, like those cars in the train. They're just not, they're not chemically connected. And that's what ligase does. So nothing is missing, no nucleotides are missing. Okay? Does that make sense? Okay, yes. It is one strand, absolutely. All you have to do is to glue them together to make it truly continuous molecule. Does that make sense? You know, like, you know Jenga? Those little sticks? You can put the sticks all together, okay? And they're going to make a continuous structure. Or you can build them up, and that's going to be continuous structure, right? But they aren't attached to each other. So you can disassemble it at any time. Now imagine every time you put the next stick, you add a little bit of a glue. Then it's going to be one huge big thing. You can raise it, move it to another place. Does that make sense? You glue it all together. Right? That's why I say that's the most abstract of all those things that we talk about. It's a bingo, it doesn't fall apart. Because everything is copied here. Lagging strand is an entire copy of the original one. Okay? But if they will separate, they're going to be like little fragments falling off.
Yeah, so you get rid of that primer, it leaves that gap.
Gap is going to be filled by DNA polymerase 1. Trying to see if we have DNA polymerase 1 here. You see how it fills the gap over here? Okay? So even when it fills the gap, you still have, see that little line over here? That black line represents the fact that those fragments are not glued together. They are not covalently bound. Does that make sense? So ligase provides the final touch. Okay? Does that make sense? Right? It's kind of, that's the most abstract concept that we're going to talk about. You seem a little bit bummed. No? You're good? Yes?
So when that's done, you're going to have DNA strand number 1 and DNA strand number 2, right? They're going to pair off. They're not re-zipping behind, are they?
No. Great question. Yes, great statement. So when we're done, let me try to get rid of all that crap over here. When we're done, that's good notice, Mark. This is going to be new molecule of DNA, double-stranded DNA, and this is going to be another new molecule. Is that what you're asking?
Yes.
Yeah. And each of these new molecules going to contain the old and the new strand. The old and the new strand. So that's why we call the replication semi-conservative. Okay? Yes? Oh, I thought you were talking about DNA polymerase. Why can't it start at the beginning? It just... Sure. Yeah, I can do it. Sure. All right. Let me get... Huh? Go ahead. Oh yeah, my man. Yeah, it does coil back together. It pretty much immediately coil. Oh, you mean like tangles and stuff? Well, yeah. So DNA doesn't remain straight. Once they replicate it, they all get coiled around and so on. Okay, so review, and I'm going to go slow, and you will tell me if something is unclear. Agreed? Topoisomerase, oh for God's sakes, topoisomerase or gyrase, okay, keeps the DNA molecule still double-stranded, straightened up, so that helicase, right here, can unzip it. When two strands are separated, sorry, in the structure that is called replication fork. they are kept separated at the fork by the single strand binding proteins. Okay? DNA polymerase number 3 recognizes the primer that was originally placed there by the enzyme called primase. Okay, it was synthesized by primase. So DNA polymerase 3 recognizes the complex between the template and the primer. It happened somewhere here. So that was still a template, and this green stuff is the primer. Okay? Does that make sense? Recognizes the complex and starts to copy. And on the leading strand, the copying is continuous. The red stuff, the red line here, on the leading strand, the copy is continuous. Does that make sense? On the lagging strand, the conditions are slightly different. DNA polymerase can read the sequence of the template only from 3' to 5'. So instead of moving in the uninterrupted fashion, like on the leading strand, DNA polymerase 3 has to run in the opposite direction from the direction of replication fork. Run away from replication fork. Then jump back and make another Okazaki fragment. And yep. Huh? It just goes on and on and on and on until the entire replication is done. Yeah, it just makes short thing, stops, jumps back. Okay? Does that make sense? Yes. That's not really, and we'll get to the proofreading and stuff, but not really, no. Okay? And it's not necessarily the same DNA polymerase molecule. It can be different molecules. Okay? Does that make sense? I mean, when we say jump, it just it makes it easier to understand. Okay? Yes. So they make short fragments because that's the only. Imagine this. Somebody walks down the hallway this way. Okay? And what I do, I walk this way, then make a gigantic leap, go all the way back to the dude, go backwards, make gigantic leap, go backwards. I will cover the entire hallway, but in a very awkward way, because presumably the only direction I can go is this, not this way. That makes sense? That's what DNA polymerase 3 does. Remember that it has to start always from the primer. Okay, primer must be there. Without primer, DNA polymerase 3 cannot do squat. Okay? And then, but primer is RNA. So these RNA primers, these green fragments, they have to be removed. And that's the responsibility of DNA polymerase 1, which cuts out RNA nucleotide and puts in DNA. Okay, it substitutes them. So in the end, you have a good old Okazaki fragment, which is entirely DNA. But those fragments are not covalently bound yet. DNA ligase stitches them back together. You have one uninterrupted continuous chain. Okay? And yes. Those are going to be two new strands.
Like on that top picture, the smaller one, it's kind of confusing to me because it looks like it was all one and then it was split.
We'll get there. Thank you for pointing that out. We'll get there. That was the plan. I'm going to go one slide back where it is explained on the larger scale. Okay. Okay. Good. Now, when this is how it happens in bacteria. Okay, you see, you have one circular chromosome that is copied, and then you have two circular chromosomes which are attached like links on the chain. And topoisomerase 4 introduces the little break here and separates these two strands, these two chromosomes. Okay? Now, to Brianna's question. And what you noticed is that, look, if we will split this picture up there, okay, then on the left half, this is leading strand, and this is lagging. And on the right half, this is leading strand, and this is lagging. Before I move to the bigger picture, any idea why? Can anyone explain? Yes?
Because it always moves from 3 to 5.
It reads from 3 to 5, so go ahead. You're on the right track.
So you're saying if it's cut there, why wouldn't it? Why?
It's not physically cut. I just make a separation, but go ahead. I would start with putting 3 and 5 prime labels on these strands. So let's go ahead. Tell me, where's 5 prime?
On the top and
Mhm.
to the left.
Excellent.
Bottom to the right.
Okay.
And then the opposite corners are 3s.
Does that make sense? Now, let me go back. Mark is absolutely right. Let me go back and show you the big physically bigger picture. Now, in this picture, there's a mistake. I'm not the person who made it. It was an artist. Can you find the mistake on this picture? Perfect. Perfect. You see? The top, for some reason, has 5 prime on the left and 5 prime on the right. So if we want to correct it, this should be 3 prime. Right? And this should be 5 prime. Does that make sense? It's a pure mistake by an artist or editor, I don't know. Now, if you now, with that correction, look at the picture here. When fork is moving this way, then this will be the leading strand, right? This part of the picture, the left part of the picture, will be exactly as this one, as this picture. So here's fork moving to the left, right? That's where the fork moves. And that's where the fork moves. Does that make sense? All the same. Leading, lagging. Leading, lagging. When we start moving fork to the other side, into opposite direction, okay, then the leading strand becomes this, and the lagging strand becomes this. Does that make sense? So whether the strand is leading or lagging depends pretty much entirely the direction of the replication fork movement. And I had the picture which pretty much imagine this. You have 4 molecules of DNA polymerase that make copies. If I will represent them as the black squares, that's going to be 1 black square, okay? That's going to be another. That's going to be third, and that's going to be fourth. Okay? So you have to appreciate that each replication fork at any given time is driven by two molecules of DNA polymerase 3. Does that make sense to you? So if one DNA polymerase that produces leading strand and one that is in the given time produces the lagging, the Okazaki fragment. But since we have two replication forks, there are 4 RNA polymerases total. You can see those two replication forks right here. Does that make sense? Does that make sense?
Those forks, they started up at the top.
Yes. Don't. No, I'm trying to think. I'm trying to think of some like real life example. I actually have none. If you think about. I don't have a good band-aid, but think about the adhesive tape. Okay. Why should we think about adhesive tape if we can make one? I will try to make a chromosome, okay, out of adhesive tape. If we would have a webcam, we could actually get this on the record too, especially the part when I struggle to get adhesive tape to work. I need topoisomerase, it coils. Okay, so this is the chromosome, okay, the initial chromosome. And I'm going to start splitting it apart. I know how, but I will try. Okay. What? No, the chromosome will get. Wait a minute. I know. I think I know what you're asking. Let me finish my craft. Okay, I feel like a first grader. Okay, so you have two replication forks. Okay, and well, sorry, it didn't work as I thought, but you got the gist, right? So those two replication forks, I was supposed to get two individual circles. Okay, does that make sense? Chromosomes, aren't they two double-stranded molecules? In bacteria, simple answer is no, and there is no complicated answer. It's one circular double-stranded DNA. In eukaryotes, who have the capacity for sexual reproduction, during the very first stage of meiosis, each chromosome gets doubled. And since each chromosome, so each chromosome now consists of, it is a homologous pair, right? So I have a homologous pair, the blue from dad and red from mom, and each chromosome in that homologous pair is getting amplified. Okay, so actually, so in a sexually in organisms who have sexual reproduction, if they are diploid, they have two double-stranded molecules. You're absolutely right, even before the meiosis. Does that make sense? One from mom, one from dad. Bacteria do not have sexual reproduction, so they do not have any mom or dad stuff. Does that make sense? You saddened that they don't have moms and dads? Yeah, that's pretty sad. They're orphans. I agree with you. It's pretty sad. Yes. No questions? You were good so far? Yes. Okay. Let's go back to this one. Okay? Let me get rid of that crap, except for the labelings. So we have to correct it, okay? So on top, you have 5' on the left and 3' on the right. Okay? You with me? On the bottom, you have 5' on the right and 3' on the left. You with me so far? Good. This is the origin of replication. And the forks start to move in both directions, to the left and to the right. Just nod if you with me, because I don't know if you puzzled or you follow. Okay? You follow so far. If this is circular chromosome, this is going to be origin. But if I will turn it like this, it's still going to be origin. Does that make sense? We just put it here just for convenience. Okay? So one fork moves to the left. Another one moves to the right. Follow? Wait a minute. We're getting there. So far, you with me? Good. So what happens? DNA polymerase can read only 3 to 5'. So if the fork moves to the left, then DNA polymerase here will have no problem reading 3 to 5'. And it will be continuous. Does that make sense? If the fork moves to the right, then DNA polymerase will continuously read 3 to 5 on the opposite strand. Because strands are it's reading the blue one, yes. Strands are anti-parallel. They are anti-parallel. Can someone let Stephanie in? Thank you. Thank you. Thank you, Jim. Okay? Are you following me on this? Mm-hmm. Yes. It reads 3 to 5. Okay. I'm now. Come over here. Come over here. Can you read this page?
Yeah.
Show me the direction that you're reading. Show me your left hand. Show me your left hand. Raise it up. Raise it up. So that's left hand, okay? So Brianna reads it from left to right. Now, if I want to read this, I either have to read it right to left, never mind that it's upside down in addition to that, or I have to flip it. Does that make sense? Now look at the picture. Here it reads from 3' to 5'. In order to do the same on the opposite strand, it has to go in the opposite direction.
But I don't get it because it says 5 to 3, and it can only read
Where?
5.
Oh, no. Oh, this one? That's new strand. That's the new strand.
Oh,
That's not reading one.
see, those numbers-
Sorry. Sorry. I couldn't.
Look at the blue.
You
That
got it?
makes sense now.
Only blue. Look on the blue.
Those numbers.
Yeah.
I was confused.
I'm sorry.
Okay.
I now realize what was the
Okay, so
confusion
now I
about.
get it.
Awesome. Great. Okay? We're good? I told you that's pretty abstract. Yeah, that's... Okay, before we move to mutations. Well, we actually moved to mutations. Let me tell you something. That process, the replication, is pretty reliable. Okay? Actually, when you think about how it works, DNA polymerase doesn't only make the copy, it proofreads it. And it's really fascinating how it does. I actually don't know the chemical mechanism, but what happens there if, say, what's some markers? Let's say the blue one, A, will pair only with T. Okay, obviously, right? So, if by any accident, and accidents can happen, A, for some reason, pairs with A, DNA polymerase will check the pairing, and if it is not right, what it means not right? It's polymerase 3. Okay, polymerase 3. If it's not right, what does it mean not right? What those nucleotides, what those bases are not going to form? Those hydrogen bonds, right? They're not going to form hydrogen bonds, proper hydrogen bonds. So the structure here is going to be all messed up. If structure is messed up, it takes it away and puts the right one in. Does that make sense? It fixes its own mistakes. It proofreads it. Does that make sense? Wait, you're being too impatient. First of all, nobody's perfect. The mutation rate for DNA polymerase is about 1 per million, something like that, 1 per million nucleotides. If you think about, so genome of E. coli is 5 million nucleotides. So it's about 5 mutations, which is okay, especially if you have some proofreading stuff. Does that make sense? We're good? It's about 5 mutations. Now, I always give this, well, we'll get to the mutations actually. Okay? Most of the mutations are not caused by the mistakes during replication. They can, but not because just misreading, like genuine misreading. Usually, it's the chemicals, the mutagens, which we'll get to. Now, mutations. I will kill you if you will tell me that protein is mutated. Well, not literally kill you, but beware. Protein is not mutated. Protein cannot be mutant. DNA can have mutations. RNA can have mutations. Protein will be altered, faulty, bad, dysfunctional, call it whatever you want, but it's not mutant. Okay? Now, RNA can get mutations during the transcription, right? When DNA is being transcribed, okay? Newly synthesized RNA can have mutation or two. That happens, okay? But mostly the mutations are induced by the chemicals or physical factors. Which physical factors you know that can induce mutations? Temperature can, not too frequent radiation. All kinds of radiation, starting from UV that we get exposed to all the time, to gamma radiation, which I hope nobody gets exposed to, X-ray, which we from time to time get exposed to. So all those are mutagens. Mutations can be of different sorts. I'm going to be mostly interested in point mutations, which means only one nucleotide is being altered. First and foremost, most mutations are silent. And what it means. This was original nucleotide, original codon in the DNA, GTA. Okay? It encoded the amino acid valine. you change GTA to GTT, it still codes valine, so nothing changed in the protein structure, right? That's why it's called silent. Like if in the text, generally speaking, you substitute huge by large, the idea of the text is not going to be very much different. Does that make sense? Now the mutation is missense. Tri-C means proline. The codon, CCC, means amino acid proline. If one C gets substituted by, say in this case, adenine, then the codon becomes ACC. Just one nucleotide is changed. And proline is substituted by threonine. And trust me, proline and threonine have fundamentally different structures. So that's going to alter protein function in quite a profound way. Protein may become dysfunctional, which sometimes bad, okay? May have no effect on the phenotype if the protein is not like very important. It may have a huge effect on the phenotype. Protein may gain new function, lose old function. Okay, so changed structure will lead to the alterations in function. That makes sense? My favorite example in the field of language science, if you have read Bible, which I didn't, there's a statement that, like, not verbatim, rich person has less chances to get into heaven than the camel to go through the eye of a needle. And every time I heard this, I thought, that's kind of a crazy fantasy. Camel through the eye of a needle? I was 4 years old. I would tell you that's not happening. I mean, that's ridiculous. Okay, it turns out in the original version of Bible, it wasn't a camel, it was a rope. I mean, we usually put thread, but in this case it was a rope. But when folks ages ago were copying Bible by hand, rope was spelled very closely to camel in whatever language they did, and they read it as camel, wrote it as camel. It never occurred to them that it doesn't sound really reasonable. So since then, camel is in the Bible. Okay, so same goes, and but you know, we still kind of get a gist of the idea. Okay, same here. Changed amino acid, changed structure. Okay, maybe changed function. Now that's missense. Nonsense mutation is when the codon for amino acid, to avoid confusion, the codon for amino acid becomes a stop codon. So instead of tyrosine here, you have a stop codon. So what's going to happen to the protein? It's going to be it's called truncated, shortened. It's actually word truncated protein. It's going to get shortened, probably going to be dysfunctional. Does that make sense? Now, one thing that I'm going to introduce is the concept of the reading frame. Because without it, we won't be able to understand the frame shift mutations. Look at the sequence of the DNA. For example, here on the top. Okay? During the gene expression, the sequence is read, quote unquote read, by triplet codons. Does that make sense? 3, next 3, next 3. Does that make sense? So it's like a frame that jumps from triplet to triplet. Does that make sense? This frame that selects each next codon is called a reading frame. Does that make sense? If you would insert a couple of nucleotides right here, that was your initial reading frame up top. Now let's take a look. What's your reading frame going to be here on the bottom. The first is the same, right? Here and here. But the second becomes, well, oh wait, oh, they're going to remove it. I'm sorry. I thought they labeled. Okay. That's what I want to do. So the first is the same, but the second without TA becomes completely different. So that's number 2 and that's number 2. Compare them. Number 2 up top in the wild type sequence is GTA. Number 2 here is GCC. It's a totally different amino acid. Okay? And all subsequent frames are going to be different. Does that make sense? This is called a frame shift mutations. Now my question, how many nucleotides should you insert or delete to cause a frame shift? 1 or 2. What about 3? There will be no shift, right? You will just introduce a new amino acid. But the rest of the protein, it's still going to read in triplet frames. That makes sense? Okay. So if you're curious how we call the mutations when you insert 3, 6, 9, 12, and so on nucleotides, this is called insertion. When you just stick some new amino acids in pre-existing protein. That makes sense? The example of the mutagen is given here. So nitrous acid, or rather the nitrite ion from nitrous acid, causes the changes of cytosine to uracil. And you can imagine that if you change cytosine to uracil, okay, you're going to change JC to AU. That makes sense? You know what sucks? Does anyone know where nitrous acid and salts of nitrous acid are used? I'm going to ruin your entire breakfast. Bacon, salami, jerky, and other processed meats to retain the color. There's a study from, like multi-center study from Britain. It was published this year, I think in the spring. They found a link between the consumption of processed meats and increased risk for colon cancer. Something about 60% increased risk for colon cancer if you eat more than pretty much 2 pieces of bacon a day. Kind of sucks. Because I love bacon and I love jerky. So I decided to screw that. I'm still going to eat it. So that's one. So the residues can be modified, right? Another way to cause mutations is to stick something between the nucleotides. Between the nitrogenous bases. Look at this. This is the double strand. I just use it for illustrative purposes. So you see those pairs, they're kind of hidden inside of the double helix, and they kind of form steps like this. Okay? So if you stick something in between, like this one, the acridine molecule, then when DNA polymerase will copy the molecule, because of the confusing structure here, alterations, twisted structure, okay, distorted structure, it can either make a deletion, will not read here in the top strand, or can make insertions in a bottom strand. Okay, so it causes mutations. And I can tell you, acridine orange, that 2 examples of such agents, so-called intercalating agents. Ethidium bromide is used in the lab to visualize DNA. Always wear gloves. When you handle the reagent, when you handle the solutions that contain it, it's extremely carcinogenic, unbelievably carcinogenic. Okay. Another one is acridine orange. Also is used to visualize DNA, but mostly during microscopic procedures. You can add it to the cells, and then the DNA in the cells will fluoresce bright orange. It's really, really pretty. Okay, but it's really carcinogenic. Okay. Another example: ionizing and non-ionizing radiation. Ionizing radiation is brutal. It breaks the strand. It breaks the sugar phosphate backbone. It may cause single strand break, which is relatively easy to fix, or it may cause a double strand break, which is not that easy to fix. Does that make sense? UV light causes the formation of so-called thymine or pyrimidine dimers, mostly thymine, when two bases become stuck together, okay, and that alters the structure during the replication. Does that make sense? Now, you have to appreciate that if we wouldn't have, question? You have to appreciate if we wouldn't have the repair system, we would be screwed. Like in days. We, microbes, everything, all living organisms, well, viruses are not living, all living organisms have repair systems that repair, fix the mutations, try to fix it. You may ask why still, people have things like the radiation disease, or why mutagens say cause cancer. Okay, look, if you will put someone in the X-ray machine for 3 days, not a single repair system will be able to correct that many mutations. So it's all about the exposure. Okay. If the repair system gets overwhelmed, that's it. Does that make sense? It's all about the relation between the capacity of the repair system to fix and the exposure to the mutagens. This is the brief list of what the mutagens can do, and don't have to memorize the exact chemical details, but you have to know the categories. So nucleoside analogs, when they are inserted, like 2-aminopurin, when it's inserted instead of adenosine, it will pair not with T as adenosine usually does, but with C, so switching AT pair to GC pair. Alright? Same goes for 5-bromouracil. Okay. Now these are mutagens that you are unlikely to come across, seriously. I mean, not in the real life. On the other hand, agents that modify nucleotides, like nitrous oxide, or nitrite, the ion that I just mentioned, convert cytosine into uracil, and that in the codons may convert GC to AT. So it really sucks. Okay, and you can come across that. I told you that that's a part of the processed foods. Intercalating agents. Acridine orange and ethidium bromide, not at home, not anywhere in the regular life unless you work in the lab. But this, yeah, dihydroxy chicken wire, the polycyclic aromatic hydrocarbons, where are you going to find them? Huh? Benzene is not polycyclic. Single cycle. Where are you going to find them? Tobacco smoke and gas. Like gasoline. Well, rarely, I didn't see any people around here, inhaling gasoline in vast amounts. But smoking, yeah, oh yeah. And that's one of the most powerful carcinogens in the tobacco smoke. Okay, so it distorts the helix, so you can get insertions, you can get deletions, you can get frameshift mutations of different sorts. Ionizing radiation, you're exposed to it maybe once a year, or maybe twice a year, hopefully, not more frequent than that. Usually in the form of X-ray, or when you fly, by the way, you're also exposed to ionizing radiation. Two things. One, DNA breaks. Second, it converts GC to AT. Okay? Yes?
Why is it when you fly you're exposed to that?
You closer, I mean, if you like
You're just higher up in that.
33,000 feet up, the cosmic rays, which wavelength-wise similar to X-ray and stuff, they penetrate the fuselage of the plane and you are exposed. I think one flight, well, it's not as bad as one X-ray, but if you frequent flyer, you probably get 1 to X-ray worth of the exposure.
What if you're on like international flights that you're up there even longer?
Oh, no. I mean, if you frequent flyer, I mean, have you seen movie Up in the Air? If you haven't, watch it. Like if you, like every day you fly from LA to New York and vice versa. Okay. And last thing, UV. We talked about it, pyrimidine dimers. Okay. When DNA tries to read this, DNA polymerase 3 tries to read it, it makes mistakes, causes mutations. So I want you to know what this, or this, or this, or this can cause. So not specifics. I will not ask you what are the effects of 5-bromouracil on the DNA, but nucleotide analogs, what effect do they have? What type of mutation can they cause? Is that clear? Awesome. All right, we're done.